Chapter 21, the Digestive System. In this chapter, we'll talk about the function and process of the digestive system. We'll look a little bit at its anatomy, discuss motility and secretion, look at what regulates GI function, the process of digestion and absorption, and then we'll look at the different phases. So we have the cephalic phase, the gastric, and the intestinal phase, and then we'll end with the immune function of the GI tract. So what are the basic processes of the digestive system? So number one, we have digestion. So digestion is when we take the foods that we consume and we break it down into smaller units. And we need to do that so that we can take the food from the lumen of our GI tract and absorb it through our cells into our blood. So digestion has to happen. Again, we want to absorb the nutrients into our blood, and so absorption is another basic process. So this is when the substances from the lumen will get absorbed into the extracellular fluid. Now, motility is another important process because we want to make sure that the foods that we eat continue to move through our GI tract. And then we also have the help of secretions. So we have substances that can come from the epithelial cells. We have substances that can come from other accessory organs of the GI tract that help with the digestive process. So a little bit about anatomy, not too much because you already took an anatomy course, but we have our oral cavity made up of our mouth, our pharynx. This is where we chew. This is also where we're going to release saliva, which contains salivary amylase, which is going to digest our carbohydrates. And so in our oral cavity, the first stage of digestion occurs. And so we have physical digestion, which is the chewing, and then we also have the enzymatic, which is through that salivary amylase enzyme. Or chemical digestion, we can call it. Okay, so the next process is the esophagus. So after we chew, we swallow, the bolus of food will go down our esophagus, and then it will get into our stomach. And our stomach is divided into the fundus, the body, and the antrum, which is really the upper part, the body, and then the lower part. And this is where we're going to mix the foods with the acids and the enzymes that are released into our stomach, and we create chyme. Then contents from the stomach will go through our pyloric valve into the small intestine, which is divided into the duodenum, the jejunum, and the ileum. Now, in the small intestine, this is where the majority of digestion as well as absorption is going to take place. So digestion is really aided by enzymes released from accessory organs like the pancreas and the liver and the gallbladder. From the small intestines, the contents will move into the large intestine, which is divided into the colon and the rectum, and then it will come out the anus. So let's take a look at the GI tract wall. And our GI tract wall is made up of 4 layers. The first layer is the mucosa layer, which we are going to see here. So this is the largest layer here, and we also have the mucosa layer here. And notice, just to orient yourselves, up here would be the lumen of our GI tract. So, and then this would be our GI tract wall. And we're really wanting to absorb the nutrients from up here down into here. So the mucosa layer is the layer that's going to be closest to the lumen. And it's made up of epithelial cells. We also have modifications here, like folds and villi. And we see that in the intestine. So this is a sample of the intestinal wall, and over here we have the stomach wall. And the first thing I want you to notice is we have the same layers, but they are quite different. So the mucosa layer in the stomach looks like this. We have a lot of epithelial cells. And then the mucosa layer in the intestines, we have these villi and crypts. So think of microvilli. So their purpose is to increase surface area. Now, remember I said the small intestine is where we're going to do the majority of digestion as well as absorption. And so Fick's law tells us to increase absorption rate or diffusion rate, we want to increase surface area. And this is why we have a lot of these villi and crypts in the intestinal wall. Below the mucosa layer, we have our submucosa. And the submucosa layer is where we're going to find connective tissue as well as our blood vessels and our lymph vessels. And then we have our third layer, which is our muscularis externa layer, which is made up of smooth muscle. And then below that, we have our fourth layer, which is serosa, which is a layer of connective tissue. Now, one of the processes of the GI tract was motility. So we want to make sure that things are able to move. And to do that, we have to have contractions. There are two major types of contractions. We have tonic and phasic that occur in the GI tract. Tonic means always, so these are sustained contractions. So they're mostly contracted and then periodically they will relax. So sphincters would be a great example of this. Phasic contractions would be the opposite. So they are mostly relaxed and then they will contract in phases. So these contractions last a few seconds at a time. And there are two major types of phasic contractions. We have peristalsis and segmentation. So peristalsis is shown over here in A, and the purpose of peristalsis is to move the contents. So in this case, we're going to move the bolus of food from one side, from the left to the right. And so the contractions happen like a wave. So it will contract here, and then the bolus will get squeezed to the right, and then we'll contract the next part, and it'll get squeezed to the right, etc. So we'll see this, for example, in our esophagus. So this is why we can swallow while we're lying down, and the food will still move from our mouth into our stomach because it's the peristalsis action that's actually moving it. Then we have segmentation or segmental contractions. These are found, for example, in the intestines. And the purpose of segmental contractions is to mix the contents. So this is a great example. Here we have a tube that's filled with different color paint. And if we want to mix the contents, one way to do that is just to contract different segments of, in this case, let's say this is the GI tract. We're just going to contract different segments. And as you contract different segments, the contents get swirled around and mixed together. And that's important because if we're talking about the small intestine, we said again, a lot of digestion occurs there. And that digestion is aided by enzymes released from the pancreas and the liver via the gallbladder. And so when these enzymes get released into the small intestine, we want those enzymes to get mixed into the contents. And so these segmental contractions will help do that. It's like kneading bread almost. Okay. So another important process of the GI tract is secretion. So I've mentioned some of these secretions already. We have digestive enzymes that help the digestive process. They help break down foods. So the first digestive enzyme our foods will come across would be in our mouth, and that would be our saliva. So saliva is released by an exocrine gland, the salivary gland. We have other exocrine glands like the pancreas, which releases most of our digestive juices. But digestive enzymes are also released by epithelial cells of the mucosa layer, for example, in our stomach and the small intestine. We also have mucus that's secreted, and the mucus is really important because it provides protection. It is a physical protection so that the acidic environment of our GI tract, especially our stomach, which is very acidic, doesn't digest our own stomach. And so that the mucus layer provides a physical protection. And mucus is secreted in the stomach by mucus cells, and it's secreted into the intestines by goblet cells. And we've mentioned goblet cells before. These cells release mucus in the respiratory tract. So I've mentioned saliva already, which is, again, released by our salivary gland, which is an exocrine gland. And then bile, I've discussed or I've mentioned, is made by the liver, and it's stored in our gallbladder and helps with the process of digesting fat. It emulsifies our fat. So how is our GI tract regulated? So we have short and long reflexes that can turn on our GI tract. When we talked about the nervous system previously, we talked about our central nervous system, our peripheral nervous system, and then I mentioned the enteric nervous system, the ENS. And the enteric nervous system is basically the nervous system of our GI tract. So it's also known as the little brain. So it's basically the fact that it has its inner neurons and nervous system, that's just part of the GI tract. And so it kind of can function almost like a CNS. So they just kind of gave it this name, enteric nervous system. So our GI tract can get turned on via a short reflex. So the short reflex is basically the GI tract turning itself on when foods enter into the GI tract. And that makes sense. Once we start chewing and bringing food into our digestive system, it will notice that there's food and it will turn itself on. And so it is a very short reflex. But we also have a long reflex, and this is also known as the feed-forward reflex or the cephalic reflex because it involves our brain. And this is what you may be familiar with when you look at food or when you think about food or when you smell food. You know that your mouth starts to water and your stomach starts to growl. So food hasn't actually entered into your body, but the idea of thinking about it, you're anticipating it coming, and so the brain will send a longer reflex because it's going from the brain and down to our GI tract, so it's called the long reflex. It's going to stimulate our GI tract to turn on. Again, it's kind of, anticipating that food's coming. So you're basically turning the GI tract on so that it's already ready to go when you start eating. But whether you turn on the GI tract via the short or the long reflex, the same things happen. Okay, so you're turning on the GI tract. Okay, other important functions or processes of the GI tract is the process of digestion and absorption. So again, digestion is taking food and breaking it down into smaller components so that we can absorb them. And remember, we have our villi and crypts in the mucosa layer of our small intestine. Those are going to be really important because the small intestine is where we do the majority of absorption, and the villi and crypts will again increase surface area so that we can absorb our foods. Now, what kind of foods are we going to eat and how do we break them down? So let's start off with our carbohydrates. So we have our carbohydrates, which are made up of glucose polymers, so starches and glycogen, and we are going to break them down from our polysaccharides into our disaccharides, so di for two. And we're going to do that with the help of the enzyme amylase. So this is why the saliva that we release in our mouth, which is also known as salivary amylase, breaks down our carbohydrates. So if you put a piece of white bread on your tongue or in your mouth and you just kind of let it sit there with your saliva, you know that that white piece of bread starts to break down. And it's the enzyme that's doing it. So we have our disaccharides, our maltose, sucrose, and lactose. Then our disaccharides will get broken down into our monosaccharides. And our monosaccharides are our glucose, galactose, and fructose. And we get our breakdown of disaccharides with enzymes called disaccharidases. And so what I want you to notice is our enzymes, a lot of the times, end in the suffix ase. So we have our amylase, our disaccharidases, and specifically to break down maltose, we will use maltase. To break down sucrose, we will use the enzyme sucrase, and to break down lactose, we will use the enzyme lactase. So all three of these, maltase, sucrase, and lactase, are disaccharidases. And we know that maltose is made up of two glucose. So when you break down maltose with maltase, you get two glucoses. When you break down sucrose with sucrase, you get a glucose and a fructose. And when you break down lactose with lactase, you get a glucose and a galactose. Now that we've broken down our carbohydrates to monosaccharides, let's absorb them. Okay, so this is the lumen of our small intestine, and here's our mucosa layer, and here's our capillary because we want to absorb into the blood. So glucose and galactose actually work similarly. So glucose or galactose, depending on what we have, will actually come into our mucosa cell with the help of sodium. So this is an example. This little blue circle is an SGLT. So hopefully you remember your SGLTs, sodium glucose linked transporter. It's an example of our secondary active transporter. So as sodium is going down its concentration gradient, it is forcing glucose or galactose to come in as well. And once glucose enters, it will go out through a GLUT transporter. And our GLUTs, remember, those are examples of our facilitated diffusion. And so this is specifically a GLUT2, although just knowing that it's a GLUT is good enough. Fructose comes in through a GLUT, in this case GLUT5, and then it goes out through a GLUT. And once it goes out the mucosa cell, all of our carbohydrates, whether it's fructose, glucose, or galactose, all gets absorbed into our capillaries. So it goes into our bloodstream. So now we have digested and absorbed. Okay, so now let's talk about breaking down proteins. So when you eat a piece of steak, we are going to chew it And that process of chewing it, that physical digestion, is just not enough to break it down to the point where we can start to absorb our proteins into our body. Now, if you recall, proteins are made up of amino acids, and here we have a chain of amino acids, and they're held together by peptide bonds. So chewing, yes, helps break down proteins, but we need further help with the help of enzymes. And these enzymes are known as peptidases because they're going to break down peptide bonds. And we have specifically endopeptidases and exopeptidases. Endopeptidases are going to break peptide bonds somewhere in the middle of our peptide chain. Exopeptidases are going to break peptide bonds towards the end of our peptide chains. So here we have amino acids with our peptide bonds, and here we have an amino terminal, that's one end, and a carboxy terminal, that's another end. So let's start off with our endopeptidases. So in this example here, we have an amino acid chain with peptide bonds, and what we're going to do is we're going to come in and break the peptide bond right here in the middle of the chain. So that would be an endopeptidase. Now there are different types of endopeptidases. We have some that's secreted by the epithelial cells in the stomach, and that's known as pepsin. And then we have others that are released by the pancreas, that's known as trypsin and chymotrypsin. And so pepsin, trypsin, chymotrypsin are all examples of endopeptidases. So again, what they're going to do is they're going to break peptide bonds somewhere in the middle of our amino acid chain. Exopeptidases, think of exit or outside. They're secreted by the pancreas. The pancreas, remember, again, is a pretty important accessory organ of the GI tract, so it releases a lot of digestive juices. And so they will release carboxypeptidase and aminopeptidase. And so the exopeptidases, which break peptide bonds at the ends of our amino acid chain, remember our amino acid chain has an amino terminal and a carboxy terminal. So if we are going to come in and start breaking a peptide bond starting at the amino terminal, that enzyme is called an aminopeptidase. If, on the other hand, we come in and we break the peptide bond at the end, starting at the carboxy terminal, that is specifically done with a carboxypeptidase. So exopeptidases break peptide bonds at the ends. And because we have an amino end and a carboxy end, we specifically have an aminopeptidase and a carboxypeptidase. And all of these peptidases, whether it's endo or exo, are going to help break our protein down into single amino acids, di or tripeptides, so that we can absorb them into our body. And sometimes we don't do such a great job, so we might have a smaller peptide chain. But once we digest our protein, we want to then absorb it. So here we have our long protein chain, and then it's going to get broken down with the help of endopeptidases somewhere in the middle, exopeptidases towards the end. And let's say we did a really good job and we ended up with single amino acids. So these are individual amino acids. This is how they get absorbed. They will come in with sodium on the apical side, and on the basolateral side, they will go out by exchanging with sodium. Okay, so for single amino acids, think sodium. It will come into the cell with sodium, and then on the other side, it will exchange with sodium. Exchange means it'll go in opposite directions, and then our single amino acid will get absorbed into our bloodstream. And notice it goes to the liver because this is our hepatic portal. Our hepatic portal system that connects the GI tract to our liver. Let's say we didn't do such a great job, so we didn't get down to single amino acids. Instead, we have di and tripeptides, 2 and 3 amino acids stuck together. In that instance, think of hydrogen ions. So what's going to happen is these guys are going to come into our cell with hydrogen, and on the other side, it will exchange with hydrogen. And again, it will go into our bloodstream. Now, notice some of our di and tripeptides can get broken down into single amino acids after it actually goes into our cell as well with the help of further peptidases. Now, if we have small peptide chains, then they're too big to go through these proteins, so they will go through via transcytosis. And if you recall, transcytosis is where we endocytose on one side, take the entire vesicle to the other side, and exocytose. And again, it goes into our bloodstream, into our capillaries. And so now let's break down fats. So we ate fats, we want to digest it, and we want to absorb it. So majority of fats are triglycerides, which is made up of a glycerol backbone and 1, 2, 3 fatty acid chains, hence the tri. And so these fats, triglycerides, are going to be broken down by lipases. So think of lipids, right? Lipase, ACE for breaking down, lipid, right? Okay, so we have lipases, colipases. So these are our enzymes. And so we broke it down into a monoglyceride and 2 free fatty acid chains. However, remember, lipids do not dissolve well in water, right? So lipids are hydrophobic. And so when we bring lipids into our GI tract, which has a lot of water in it, our fat will form a big fat droplet. It won't dissolve. And so this is where bile comes in. So we know bile is made by the liver, it's stored in the gallbladder, and the bile or bile salts that are released will help emulsify our fat. So it takes this gigantic big fat droplet and it breaks it down into smaller emulsions or smaller droplets of fat. So the bile salts essentially surround the lipids into smaller little bubbles. And now that we've emulsified and kind of broken the fat into smaller pieces, the enzymes from the pancreas, the lipases, the colipases, are now able to go in there and digest our fats. And once it digests it, we create a micelle. Now that we have our broken down pieces of fat, they will get absorbed into our small intestine cell wall. So our monoglycerides and free fatty acids can enter via diffusion because these are lipids and our cell wall is a lipid bilayer, so it can kind of just go through. Cholesterol is a little bit bigger, so it's going to go through a transporter in the membrane. Once inside, the absorbed fats get combined with cholesterol and proteins. With the help of the smoothie plays a role. And by doing that, we're going to create something called a chylomicron. So that's going to be our lipids, our cholesterol, our proteins. We have this little chylomicron, a little ball of just fat and proteins, etc. And the Golgi will then package it into a vesicle. And so again, this is where the smoothie because we're talking about lipids, is going to play a role here. So now we have the smoothie the Golgi, and it's in a little vesicle, and so the vesicle will exocytose that chylomicron. And here is where it's also different. Instead of getting absorbed into our bloodstream, it actually will go into our lymphatic system. And then from the lymphatic system, it'll then drain into our circulatory system. So you can see it says lymph to our vena cava, and that's when it's now in our circulatory system. Okay, so we can also digest and absorb nucleic acids and vitamins. So nucleic acids are digested into its nitrogenous bases and monosaccharides. And the monosaccharides will be absorbed via facilitated diffusion, like we said earlier about how monosaccharides are absorbed. And then vitamins and minerals are absorbed in the intestine. So some of them are fat-soluble vitamins like vitamin A, D, E, and K. So they're absorbed in the small intestine along with fat. And then the water-soluble vitamins like C and most Bs are absorbed via mediated transport. One vitamin I also want to point out is vitamin B12, and we did mention this when we were talking about anemia. So vitamin B12 is necessary to make red blood cells, and so when you have a lack of vitamin B12, you can become anemic. And one specific type of anemia was known as pernicious anemia, and that's because vitamin B12 needs intrinsic factor to be absorbed. Intrinsic factor is made by the parietal cells in the stomach, and so if the parietal cells in the stomach are not able to make intrinsic factor, then you're not able to absorb vitamin B12, and so you can be anemic. And if you have anemia because of that, because of not having enough intrinsic factor, then what you have is specifically called pernicious anemia. Okay, here is a nice summary slide. And what this does very well is it tells us the type of motility, secretion, digestion, and absorption that happens in different parts of our GI tract. For example, in the oral cavity and the esophagus, what kind of motility do we have? We have the process of swallowing and chewing, and obviously the peristalsis going down the esophagus. What kind of secretions do we have? We have saliva, right? Our salivary gland will release that. And we also release some lipases here. So then what kind of digestion do we do here? Well, the salivary amylase will digest our carbohydrates, and the lipases will digest our fats, although again, it's minimal here. And do we do any absorption here? And the answer is none. And then you can see the same goes for the stomach, the small intestine, and the large intestine. So it just summarizes for you the different things that are happening in the different areas. So use this as a summary slide. So hopefully you'll come back after you've gone through the GI tract and review. What I want to do is talk about the different phases of the GI tract. And so then we'll go more into what happens, for example, in the stomach, okay? So let's start off with the first phase, which is the cephalic phase. So we talked about the word cephalic, so you should think of our brain. But here, the cephalic phase is actually everything above the stomach. So yes, thinking about food, smelling food, looking at food, all of that is part of the cephalic phase. Remember that feed forward where we can think about food and it can turn on our GI tract. That will all be still part of our cephalic phase. But so is food entering into our mouth, chewing, swallowing, all of that is still part of the cephalic. So everything above the stomach is the cephalic phase. Chemical and mechanical digestion will occur. Again, that starts in the mouth. We'll release saliva, which helps soften and lubricate the food. And then again, we have some chemical digestion happening here with the amylase and some lipases. And then we have the process of chewing, mastication, that helps also break down our foods. So let's talk about swallowing. So swallowing occurs as a reflex, and it occurs when food gets moved down, obviously, to the back of our mouth, and it triggers that swallowing reflex so that we can swallow down the esophagus. So the first thing I'd like you to notice is our esophagus over here, and then the trachea over here. So the trachea is the opening to our airway, or this is where air is going to flow. And so remember, our respiratory tract is, or branches, tubes, are surrounded by cartilage, and that helps keep it open. And so you can see how our respiratory pathway is open. And so this allows us to breathe and air to move easily. So normally we have a tonically contracted upper esophageal sphincter. So this sphincter up here in the esophagus is tonically closed so that when we breathe, air will go down into our trachea. But sometimes we want to swallow our foods, and so that triggers a swallowing reflex. And basically what's going to happen is this upper esophageal sphincter will relax, and that will allow it to open so that the food can go down the esophagus. And also we will close the opening to the trachea. And so the epiglottis will make sure that the food goes into our esophagus and not into our airway. And then with the help of peristalsis, we will move the bolus of food down into our stomach. So this is a swallowing reflex. So once the bolus of food enters our stomach, we are now into a new phase. That is our gastric phase, gastric for stomach. And so the stomach has three general functions. Number one is we store food here. This is why you can eat a lot of food and your stomach gets really big because it's able to expand and hold food. So it stores food here, and that's important because we want to regulate the passage of food into the small intestine and really not overwhelm the small intestine, because again, the small intestine is where we're going to do majority of our digestion and absorption. So we want to make sure that we are passing food in amounts that the small intestine can handle. Another function of the stomach is digestion. So we can chemically digest foods as well as mechanically through the churning that happens in there. And the chemical digestion happens with the acidic environment. It's very acidic. It has a pH of 2 with the enzymes that are also released in here. And then the stomach also helps protect us. So it protects the body by destroying any bacteria or other pathogens that might have been swallowed with the foods that we eat. Now, obviously, it's not perfect. For example, H. pylori is a bacteria that can survive in our stomach, even though it's very acidic. So it can't protect us from everything, but it does protect us from a good amount of things. So for the stomach, remember the mucosa layer of the stomach? We saw an example of that earlier. We said it has a lot of epithelial cells. And these epithelial cells are able to make substances and secrete it. And so this is a good table that tells you all of the different cell types in the mucosa. Okay, the gastric mucosa, so the mucosa layer of our stomach. So for example, we have our mucus cells, which secrete mucus and bicarb. We have our parietal cells, which secretes hydrochloric acid and intrinsic factor. We have our ECL cells, which secretes histamine, our chief cells that secrete pepsin and lipases, our D cells, which secrete somatostatin, and our G cells that secrete gastrin. So here are the different cell types, what they secrete, what stimulates those secretions to be secreted, to be released, and then what those secretions do. So please use this as a review slide, a summary slide, a study slide, but I'm going to go over it with this slide because I like the figures better. Okay, so however we turn on the stomach, okay, whether it's through the food coming in, which would be again a short reflex, or through higher input via the vagus nerve. Whatever we're going to do, whether it's short or long reflex that turns our stomach on, it all starts with stimulating our G cells. So our G cells will secrete gastrin. Easy to remember, G-gastrin, GG. And think of this as your go signal, okay? So the G cells secrete gastrin. It's like our go signal. It turns our stomach on. And so our stomach is filled with acid. It's filled with hydrochloric acid. And the cell that releases that is your parietal cell. So if we're going to turn our stomach on, that's what we want to do. So we want our G cells to secrete gastrin to go, right, to tell our stomach go, work. And so if you follow the line, it's going to stimulate our parietal cells to release hydrochloric acid. Okay, so here we have a hydrogen ion, but it will combine with chloride and be hydrochloric acid. So it's a very acidic environment. Now, this is really what we need to happen, right? We need hydrochloric acid to be released into our stomach. To really make sure this happens, the gastrin not only directly stimulates the parietal cells to do that, but it also stimulates the ECL cells to release histamine, and it wants the histamine to also tell the parietal cells to release hydrochloric acid. So it's like I don't know, kind of like insurance. So gastrin is going to directly tell the parietal cell release the acid, but just to make sure, I'm also going to stimulate the ECL cells to release histamine, which will also stimulate the parietal cells to release hydrochloric acid. So now the acid level in the stomach is going to rise and rise and rise. That's going to stimulate the chief cells. And the chief cells will release pepsinogen, which turns into pepsin, which digests our proteins. It will also release our gastric lipases, lipids, which helps digest our fats. And so what we've done now is we've just really turned our stomach on. We have increased the acids, the hydrochloric acids, and we've released enzymes. Now, eventually we have negative feedback. So once your acid level starts to rise too much, we want to be like, okay, that's enough. We have a good amount of acid, so let's turn this whole thing off. And that negative feedback pathway will stimulate our D cell. So think of the D cell as your done signal. And the D cells will release somatostatin. And we've heard of somatostatin before when we were talking about the release of growth hormone. And I think also when we were talking about the pancreas and the release of insulin and glucagon. Remember in the pancreas, we had the D cells releasing somatostatin there as well. But it always did the same thing. It basically shuts everybody off. And that's what it's going to do here as well. So somatostatin released by the D cell is your done signal or your stop signal. So easy to remember, D for done, S for stop. And so it's going to stop everybody. It's going to tell the G cell to stop, so it'll negatively impact that, right? Negative feedback onto that. It will tell our ECL cell to stop. It'll tell our parietal cells to stop and our chief cells to stop. So everyone stops and you're done. So let's do that one more time very quickly. So whether it's food directly entering into our stomach or higher input, we're going to turn on our G cells. G cells will release gastrin. It's our go signal. And so it will tell our parietal cells to go and release our hydrochloric acid. Just to make sure, it will also stimulate our ECL cells, which releases histamine, and that will also tell our parietal cells to release hydrochloric acid. The buildup of acid will stimulate our chief cells to release pepsinogen, which turns into pepsin, as well as gastric lipases. And then eventually we need to say, okay, we're done. Let's shut everybody off. And that will be done by stimulating our D cells. Our D cells will release somatostatin, and that will be our stop signal. And it'll stop everybody. It'll stop our G cells, our ECL cells, our parietal cells, and our chief cells. Now, there are a couple of things that we're missing here, and you can look back at that table and add them in here. One thing I want to point out is the fact that our parietal cells also release intrinsic factor. Remember, I just talked about that when we were talking about vitamin B12 absorption. So you can add that here just to have it all in the same figure. And another thing that's not listed here is your mucus cells and the fact that they release mucus as well as bicarbonate ions. And it's not on this figure, it's on the next one. But again, if you want everything in one slide, you can add it in. So how then, if our stomach is so acidic, if it has a pH of 2, how does our stomach not end up eating itself? And the reason is because we have protection. We have both a physical protection as well as a chemical protection. Our physical protection is made by the mucus layer. So the mucus cells secrete mucus, and it literally creates a barrier so that the stomach juices can't get to our GI tract wall. But just in case, the mucus cells will also release bicarbonate ion. And remember, bicarbonate ion is a very strong buffer. So if any of the acids gets through the mucus layer, let's say we have an area of the stomach wall where the mucus layer has thinned out, we have bicarbonate ions there as well, ready and waiting to buffer any of the acids. So if everything is working right, the physical protection, the mucus, and the chemical protection, the bicarbonate ions, will be enough to protect our stomach wall. Now, sometimes things don't work right. That's why you can get ulcers. Okay, so that's the stomach. Now let's move on to the small intestines. And so once we move on to the small intestines and then the large intestine, we are in the intestinal phase. So the intestinal phase is going to occur again when food enters into the small intestine, and it's going to cause the release of the enzymes from our pancreas to help release the enzymes necessary to digest and absorb the foods that have come into the small intestine. And we've already talked about those when we talked about the digestion of our proteins, our fats, and our carbohydrates. Okay, so here are some examples of the enzymes that are released into the intestine. Remember, the pancreas is a really important accessory organ, so a lot of our enzymes come from the pancreas. Those include our trypsinogen, our chymotrypsinogen, pro-carboxypeptidase, pro-colipase, and pro-phospholipases. Now, notice these have the word pro in it, and if you remember, when we were making our hormones, we had our pre-pro hormone, etc., and then it turned into our pro hormone. And remember, it meant that that hormone was not quite ready yet. So these are the same, this is the same concept. So these enzymes, what they're telling you is they're being released in a inactive form. And so it's inactive in the pancreas and released that way into the small intestine. And once it gets entered into the small intestines, it will get activated here. And that's because if these enzymes were activated within the within the pancreas, it will actually auto-digest the pancreas, which we don't want to happen. So the pancreas will make it up to like almost ready, so it's still inactive. We need one more final step, and that final step to activate it doesn't happen until it's released into the small intestine. Also, don't forget, we have our bile released, made by the liver and stored in the gallbladder and then released into the small intestines. Remember, that helps with digestion of fat by emulsifying our big fat droplet. And then here we have our goblet cells. So the goblet cells release mucus to, again, provide that protection from our GI tract wall eating itself up. And the hepatic portal system is our third portal system. So we talked about the hypothalamic hypophyseal portal system which connects the hypothalamus to the anterior pituitary. We talked about the renal portal system, which connects our glomerulus capillary to the peritubular capillary in the nephrons. And then we have our hepatic portal system. And the hepatic portal system connects the capillaries in the GI tract to the capillaries in the liver. So most fluid is absorbed in the small intestine, or most of the foods that we eat we're going to, again, absorb there. And this portal system allows us to take the foods that we have absorbed and take it to the liver. And the liver will be able to kind of clean up some of the contents that we have absorbed. And we want to do that before we send the blood to the heart, because once it goes to the heart is going to pump it everywhere, including our brain. And so this is why, think about people who drink a lot of alcohol. One of the organs that really gets negatively impacted by that is the liver, because we're absorbing in the GI tract a lot of alcohol. And so the liver is having to kind of deal with all of that alcohol. Okay, what else happens in the intestinal phase? So again, just to remind you, most digestion occurs in the small intestine, as well as most of our absorption. So both digestion and absorption occurs here in the small intestine. The large intestine is really important for concentrating our feces. So what it does is it will do our final absorption of water. So most of our water will be absorbed in the small intestine, but that final last bit is going to occur here in the large intestines. So if you were to move the contents too quickly, you don't have time to absorb the water, so that's how you get diarrhea. And then finally, the immune system plays an important role in our GI tract. So we have these cells called M cells, and what they do is they sample the contents of our gut, and if they notice anything that's there that shouldn't be there, it will stimulate an immune response. So these immune cells, these M cells, will secrete cytokines, which triggers an immune response, an inflammatory response. Now, that's once the food or the pathogens have entered into our GI tract. But let's say we just ate something that was toxic, then we're going to trigger a vomiting reflex, which is protective, so that we vomit up the contents before it goes any further into our GI tract. And that is our digestive system. Thank you.